Hello to all the HubLE audience. My name is Fatemeh Malekipour. I am a postdoctoral research fellow at the University of Melbourne, Australia. And I'm also a member of Meet the First Author committee at HubLE. I'm here today for an edition of the Meet the First Author with Judith Piet, if that's the wrong, correct?
Yeah.
Great. We will be talking about their paper titled Marrow Aspiration in Aged Mice, Intermedullary Osteogenesis, Reduced Mechanoadaptation, Increased Marrow Fat, which was published in Connective Tissue Research Journal. So Judith, welcome to HubLE. And to start with, could you tell us the summary of your work and how it fits into the big picture of your field?
Okay. So this is work that I conducted in the context of my PhD with Dr. Scheffelbein at Northeastern in Boston and with Professor Barron at the Harvard School of Dental Medicine in Boston as well, and I did my histomorphometry with him. So to explain in the bigger context, my PhD was focused on how to increase mechanoadaptation in old mice in their bones. So mechanoadaptation is the fact that bones adapt to external loads, and we know that it is decreased with age. So what I did is I used a model of in vivo tibial loading. So in mice, you can fix their tibia in little loading cups and apply cyclic compressive loads. If you do that 3 times a week for 2 weeks, it's enough to increase bone formation in the loaded leg compared to the contralateral leg. But we know that this response is decreased with age. So the idea of this specific paper was to prime the bone to be more responsive to loading using marrow aspiration. And the reason why we thought of using marrow aspiration is because in young mice, it's known to be osteogenic. So if you aspirate the marrow, you're going to have a regeneration sequence with first progenitors coming to form bone in the medullary cavity, and eventually that bone is resorbed. So our idea was, can we prime the bone with cells to be more responsive to loading using marrow aspiration? So the paper had 2 objectives. The first one was to assess the effects of marrow aspiration in old mice, because it hasn't been before. So in terms of where is bone formed and marrow adiposity. And the second objective was to see the effect of marrow aspiration on mechanoadaptation. So combining aspiration and loading compared to loading only. So what we found is that in these old mice, when you aspirate the marrow, you have a transient increase in the amount of bone. So with that, we did that in the tibia. So the aspirated tibia has transiently more bone than the contralateral tibia, and it's mainly caused by intramedullary bone formation. But it doesn't last, and importantly, we found that marrow adiposity was increased eventually in the aspirated tibia. Now, when we used histomorphometry to look into more detail at where bone was actually forming, we found that with aspiration, you have mainly bone formation on non-periosteal places. So the endosteal surface, intramedullary, intracortical remodeling, etc. When you have loading only without aspiration, we found what we had found in previous papers, that you have some formation endosteally and periosteally. And now when you combine aspiration and loading, the response to loading is actually decreased compared to loading only. So it's going against our hypothesis that actually combining aspiration and loading is going to further decrease mechanoadaptation in these old mice. And we also find what is quite interesting, that in mice that had aspirated marrow in their right tibia, periosteal formation was also decreased in the contralateral tibia compared to mice that did not have aspiration. So it suggests that there's kind of a systemic decrease in bone formation in these mice. Yeah.
Okay, that's very interesting. So, I mean, we basically know all the effects of mechanical loading, but now you have sort of combined the effect of fat tissue, if I put it in a, is that right? In a simple way, or?
Yeah, so further research would be needed to actually elucidate the mechanism, what we can see here is that there's a coincidence between increased adiposity and decreased mechanoadaptation, but we don't have a causality, but it's interesting to observe because previous research has highlighted that increased adiposity can be linked to decreased skeletal health. So that's an interesting finding. And also it can inform, it might inform therapies in older people that have marrow injury for their skeletal health.
Okay, that's great. So if you want to put it in basically what's the most important thing that you learned from this study, how would you put it?
Marrow aspiration further decreases mechanoadaptation. So it's not a good tool to increase adaptation in these old mice. Yeah.
Right. Okay. Great. Great. So what was the greatest challenge basically that you had during the study? I suppose working with small animals like mice is not already very easy, but what was your challenges and how did you overcome them?
Yeah. So I had worked with mice before in previous papers. So in this paper, we used old mice that are about 2 years old. So it's an advanced age. So we have to buy the mice young and then we age them for more than a year. So there are two challenges. The first one is that they start dying from old age, unrelated to the treatment. So it causes a problem in sample size. And also their response at 2 years of age are very variable. So I did research with tibial loading in younger mice and in older mice. And in older mice, there's just much more variation. And with the aspiration, you have even more variable responses. So we had to be smart about how to allocate the sample size in the different groups and plan that ahead. So yeah, ideally we would do the experiment with even more mice, but down the line we realized that some mice had died, and yeah.
Okay, great. And what was your experience publishing in Connective Tissue Research Journal? How did you go with that?
Yeah, so it was, obviously it was successful because we did publish.
Yeah.
But yeah, it was a very,
in terms of
yeah, it was a very smooth process. So it took maybe 2 or 3 months between the first draft and then the accepted paper. So we had 3 reviewers, 2 rounds of revisions, but I found it very straightforward, and yeah, it was a very good experience overall.
Okay, great. So what do you expect the outcomes of this work to be in terms of clinical or research trials, or I don't know, patients, anything?
Yeah, so I think it's, for now it's far away from clinical research, but if I had the opportunity to keep working on that specific project, because now I have moved away from the lab, I would be very interested to look into, first of all, cellular senescence. So is cellular senescence increased in the bone after marrow aspiration? Could it be a reason why mechanoadaptation is decreased? I would be very interested to look into the connection of increased adiposity and decreased mechanoadaptation, because I think that's a very, it's a hot topic, and it's very interesting to see that correspondence. I would also be interested to look at that systemic effect. So we see that even in the bone that was not aspirated, we have decreased periosteal formation. Some previous literature has shown that in younger rodents, in mice and rats, you have systemic increase in bone formation, but what we see is that actually periosteal formation is decreased in other bones. So that's very intriguing. So if I had the chance, I would look into that. Yeah.
Mm-hmm. Great. And I guess the final question is, in terms of your research career and future goals, how will this publication actually help you achieve those aims?
So yeah, so I'm very interested in the mechanics of the musculoskeletal system in general. So now I have moved to another lab where I keep looking at mechanics of the musculoskeletal system. So I'm now in Belgium at the KU Leuven in the group of Ilse Jonkers.
Mm-hmm.
And I've moved from bones in mice to cartilage in rats. So my new project is looking into building a multi-scale adaptive model of cartilage in the rat knee to look at how destabilization of the knee can create an environment that disrupts cartilage homeostasis. So I think from that paper that we just talked about, I learned a lot of tools about working with animals, how to analyze the samples, the different techniques to analyze the bones, etc. So now I'm hoping to move more into modeling, which is why I joined the group in Belgium. But I think I learned a lot during my PhD in terms of animal research. Yeah.
Oh, that's awesome. So you have moved from bone to cartilage, and I suppose if you want to look at the osteoarthritis and that kind of musculoskeletal diseases, this is the best approach because they are very integrated. And
yes, yeah. And I'm also interested in the bone cartilage interface. So what happens in subchondral bone, and bone will stay around, I think.
Yeah, that's great. Awesome. Thanks for your time today, and we would definitely be looking forward to more of your work in rats and cartilage, as well as your current papers. And I wish you all the best. I would also like to thank the audience for taking the time to join us today. And if you would like to be involved in the Meet the First Author initiative, please feel free to contact us. That's it, and enjoy the rest of your day. Thank you very much.
Thank you very much. Thank you.